So our next speaker is Kathy Cullingham, and Kathy's going to talk to us about differential introgression of putative adaptive loci across the mosaic lodgepole jack pine hybrid zone. Where's the full screen? Just the green one. Technology. It's almost there. All right, well, hybrids are fascinating and hybrid zones are fascinating. Hybrid zones allow introgression of loci from one species into another. And sometimes those loci might be adaptive in the parental species and also become adaptive in the recipient species. And sometimes they might be neutral in the parent species and become adaptive in the recipient species. And the hybrid zone I'm really interested in is the lodgepole jack pine hybrid zone that goes through Alberta, British Columbia, and the Northwest Territories in Canada. And I've been working with a graduate student, Dania Remchak, on the question of how climate interacts with differential introgression in this hybrid zone. And so to answer this question, we've got a slightly complex workflow that I've tried to break down into beautiful little pictographs. And so we started with a pretty large data set. So we have over 1,300 individuals genotyped at over 10,000 SNP loci. And those are SNP loci that were from the AdaptTree SNP chip. And we broke those into separate groups based on their species ID. So we ran them through structure to get at who was a hybrid, who was pure lodgepole, and who was jack pine. And then we took the lodgepole pine that was pure, and we looked for population structure, and we looked for SNPs that were associated with climate that suggest that they might be adaptive to those climate conditions. We did the same thing for jack pine, and then we subset our data, and we subset it into different hybrid zones and the parental classes only for the loci that had climate associated SNPs. And then we examine these climate-associated SNPs using introgress and looking at the differential patterns of introgression between lodgepole and jack pine in these hybrid zones. And then following that, we looked at the allele frequencies of these interesting loci across the entire landscape. So we used the full data set to make pretty pictures, because pretty pictures really do tell a story. And so for our species ID from our over 1,300 samples, we had 540 lodgepole pine, 669 jack pine, and 169 hybrids. And these are mapped on the landscape that you see here. And then from that, we didn't find any population structure in lodgepole or jack pine. We weren't surprised by this. But when we did the climate analysis and looked at SNPs associated with climate, we found 156 SNPs in lodgepole pine. And they were associated with things like elevation, mean annual temperature, temperature difference, and our H, which is relative humidity. And then for jack pine, we found some similar environmental associations, but in addition, we found number of frost-free days is really important, as well as climate moisture index and extreme temperature. So from that, we took these markers, so the 156 SNPs in lodgepole pine that were associated with climate and the 189 SNPs in jack pine, and looked at whether or not they were differentially introgressed. And so we separated the data set into a northern set and a southern set of hybrid zones. And I'm going to call it the northern extent, and those are in pink symbols, and the southern extent in blue. And you might say, well, there's trees beyond those pink symbols, but if you're actually in the Northwest Territories, good luck finding a tree in those locations. So if you found a tree, you're lucky, but that really is as far north as the tree distribution goes. And so we examined patterns of introgression between the blue and the red that were different. So we looked at the northern, we looked at the southern, and compared their patterns. And so this is even more complex, but I can break it down for you. So on the x-axis is the hybrid index. So 0 means they're jack pine, 1 means they're lodgepole, 0.5 means they're hybrid. On the y-axis on your left, that's the probability of the genotype, with 1 being the probability of the jack pine homozygote, 0 being the probability of the lodgepole pine homozygote, and 0.5 being a hybrid. And then we have the southern cline and the northern cline. And on the other y-axis, we have the number of observed individuals with those particular genotypes. And so I've pulled one of the markers that was significant in the north and the south. And when I say significant, there is a null expectation of what you would expect for introgression based on the parental allele frequencies. And this is in those dark green color blocks. And in the light green color block is the null expectation for a heterozygote genotype. And the solid line is our actual observations. And so when that deviates from that solid green, it tells us there's something different going on here, than it's not neutral. And the dotted line is our neutral expectations for our hybrid genotypes. So for this example here, we noted that in the south, we found more jack pine homozygotes and more heterozygotes with the jack pine homozygote than we would expect based on those parental allele frequencies. And in the south, we find less than. So it tells us that, maybe in the north, where we have less, maybe it's not as adaptive in the north, and in the south, it's more adaptive. And so it's being selected for. And so we've found 28 markers of those climate-associated SNPs that showed differential patterns between the north and the south hybrid zones. And I've pulled a couple of examples to give you an idea of what sort of patterns we're seeing and what they might mean. So this pattern we observed in a number of loci, and this locus was identified in lodgepole pine, and it was associated with temperature difference or continentality. And what we found here with the clines was that the jack pine associated allele was introgressed in the northern hybrid zone. So it was introgressed into lodgepole pine. So we found a lot of jack pine type homozygotes in lodgepole pine. And you can see the red and yellow is really present in that northern hybrid zone and not in the southern hybrid zone. And we looked at the annotation thanks to Rhiannon's bioinformatic wizardry. And this locus is potentially associated with RNA processing and stress response. Another pattern that we saw quite often, and this was identified in jack pine, and it was associated with the number of frost-free days. And in this example, the lodgepole allele, so that blue color, is introgressed in the northern jack pine more than you would expect based on neutral expectations. But in the south, the jack pine allele actually looked like it was selected for in jack pine. So in the south, that allele is really important in jack pine, but in the north, the lodgepole pine allele is more important. And this locus is potentially associated with protein homeostasis, which could do all sorts of cool things. We can make up fun stories when it comes to annotation. And then the third example, this one was pretty fun. It was a locus again identified in jack pine, but associated with climate moisture index. And in this one, heterozygotes observed more frequently in the hybrid zone in both the north and the south. But it was really interesting to me because we see that the blue color is in both parentals, and the blue color means pretty much 0. And so this suggests that there's some sort of heterozygote advantage in that hybrid zone for this locus. And again, it's associated with signal transduction and perception. I mean, come on, let's drool over these annotations because they're so much fun. And so from this work, we're still kind of unpacking it and interpreting all of these clinal zones. But what we found is that hybrid zones are still really fascinating, and that we found some contrasting environmental differences between lodgepole and jack pines. So there's different environmental variables that are important in their adaptation, and that there is evidence of differential introgression in this hybrid zone, and some of it is potentially associated with climate. And these sorts of information can be really important for understanding climate change impacts, forestry, reseeding, stock management, all of those really great things that we're interested in. And with that, I want to say thank you.
We have time for a question for Poppy. Yeah, what's the range of genomic proportions between lodgepole and jack pine in the hybrid zone among individual trees?
It varies.
How does it vary?
It's a lot of it's, I would say it varies with the environment. And so in some regions, you'll find more lodgepole-like hybrids, and in some regions, you'll find more jack pine-like hybrids. There's a really strong environmental effect on what kinds of hybrids you're going to find.
If you find one that's more jack pine-like, its neighbors probably going to be similar.
Yeah.
And then that would be like 50-50, 25-75. What's sort of the proportional ranges?
It really varies. There are some really strongly back-crossed individuals, and we have detected F2s as well. So we've kind of got a good range of hybridity among our individuals.
Do you get F1s every generation?
We get F1s really rarely.
Oh, rarely? Yeah.
Rarely, yeah. In our samples so far. We're searching for them because that's what I want more of those hybrids in these analyses. Yeah. Thanks.